Hello everyone, my name is Kiran Chaudhary and I am your botany faculty at Infinity Learn. My dear students, आज के इस वीडियो में हम लोग डिस्कस करेंगे कंप्लीट चैप्टर सेल साइकिल एंड सेल डिवीजन. Not to forget, every year NTA is asking at least 4 questions from this chapter. चैप्टर छोटा है, बेहद कॉन्सेप्चुअल है और बहुत ज़्यादा क्वेश्चंस 4 questions, 4 questions is quite a high number when we talk about the NEET exam. And remember, some questions related to this chapter you may find in the botany section as well as in the zoology section, my dear students, in your coming NEET exam, right? This is the pattern what NTA is following from the last 2 years. So chapter is important, conceptual, as well as it has a high weightage with respect to your NEET exam. So let's get started, my dear students. Cell cycle and cell division. सबसे पहला टर्म जो आप लोगों को सीखना है, remember in this lecture I will be teaching you the entire concept, the entire chapter, and we will be mainly focusing on those concepts, on those facts and points जिस पर NTA has already asked question and there's a high probability it might ask question in the coming NEET 2023 as well. तो सबसे पहला जो चीज़ आपको पढ़नी है वो है cell cycle. So what is a cell cycle? Listen this very carefully. Cell cycle is a sequence of events where a cell is going to replicate its genome, is going to produce other constituents of the cell, will prepare for the division and eventually divide to give rise to daughter cell. तो इसका मतलब सेल साइकिल आप उन सभी इवेंट्स के बारे में बात कर रहे हैं जो सारे के सारे सेल के अंदर होते हैं बिफोर अ सेल कैन डिवाइड इनटू डॉटर सेल्स. तो देखिए इस तरीके की नजर आती है आपकी सेल साइकिल. सेल साइकिल इज एक्चुअली डिवाइडेड इनटू ब्रॉडली डिवाइडेड इनटू 2 phases, इंटरफेज एंड एम फेज. तो ये आपका यहां से लेकर यहां तक, दिस इज योर इंटरफेज. And remember, the area in the circle that you can see, my dear students, is corresponding to the time, right? तो एक cell cycle के अंदर जब भी आप cell undergo करती है through a cell cycle during cell division, my dear students, maximum time a cell is going to spend in interphase, and that is why interphase is also known as the preparatory phase. Because for example, एक analogy draw कर सकते हैं, आप लोग NEET exam की तैयारी कर रहे हैं, you might be preparing from 1 year, maybe 2 years. So आपका preparatory time 1 year या 2 year का होता है, लेकिन आपका actual exam तो सिर्फ 3 hours का होता है and 3 hours and 20 minutes right now, right? So इतना कम टाइम होता है मेन एग्जाम का. उसी तरीके से very less time a cell takes to actually divide in the M phase. So this is your M phase, my dear students, where a cell actually divides and it is quite shorter in comparison to interphase, which is the preparatory phase. तो ये आपका सेल साइकिल है जिसमें G1 है, S है, G2 है इंटरफेज में, और M फेज में आपका आएगा karyokinesis. These all the stages corresponds to karyokinesis and this is your cytokinesis. Eventually the cell, the parent cell is going to divide into daughter cells. तो इन सारे पॉइंट्स को हम लोग काफ़ी डिटेल में डिस्कस करेंगे, डोंट वरी. So this is your cell cycle, sequence of events, and remember ये सारे के सारे जो सीक्वेंस होते हैं they are under genetic control because even a single mistake can lead to abnormal formation of daughter cells which is not required, not desirable. Next, तो अगर हम बात करें ड्यूरेशन सेल साइकिल की मेरे प्यारे बच्चों, सो डिफरेंट सेल्स, डिफरेंट ऑर्गेनिजम्स का जो सेल साइकिल का ड्यूरेशन होता है वो वेरी कर सकता है ऑर्गेनिज्म टू ऑर्गेनिज्म, और विद इन एन ऑर्गेनिज्म सेल टू सेल भी वेरी कर सकता है. 2 इंपॉर्टेंट फैक्ट है जो एनसीईआरटी में दे रखे हैं, यू गॉट टू रिमेंबर दैट कि एक यीस्ट सेल को डिवाइड करने के लिए सिर्फ 90 मिनट्स लगते हैं. सो वंस इन 90 मिनट्स अ यीस्ट सेल इज गोइंग टू डिवाइड, राइट? एंड फॉर अ ह्यूमन सेल, my dear students, a human cell takes 24 hours to divide. And not to forget, M phase will last for only about an hour. यानी 23 hours आपका इंटरफेस चलेगा, preparatory phase चलेगा. Important fact है, याद रखिएगा. Next thing that we need to discuss is, my dear students, the phases in a cell cycle. I have already explained you, I have already given you the introduction. Now let us talk about them in a bit detail. The cell cycle is broadly divided into 2 phases, interphase and M phase. Interphase is the preparatory phase. A cell is going to prepare for its division, right? And M phase is the phase where actual cell division will take place. Actual division will take place, and here only preparation will take place. तो देखते हैं किस टाइप के events होते हैं during interphase. First important fact about interphase is it is between two successive M phase. तो आप देख सकते हैं, to understand this point you have to imagine two cell cycle. So this is one cell cycle and this is your interphase. ये एक cell cycle का interphase है and this is the M phase. Now imagine another, my dear students, cell cycle. So when you keep two cell cycles in mind, you can say easily that yes, interphase is actually between two successive M phases. Next thing is, it is the longest part of the cell cycle. Very important fact, and I have right now explained you with the facts that a human cell को डिवाइड करने के लिए 24 आवर्स का टाइम लगता है और इसके अंदर 23 आवर्स रफली सिर्फ प्रिपरेशन में जाते हैं, इंटरफेस के लिए जाते हैं. Interphase lasts more than 95% of the duration of the cell. This point basically explains the second point. So these are important lines, words from NCERT that you got to pay attention, and you can expect a question in your coming NEET 2023 exam. Next, this phase, this interphase is known as resting phase. Now everyone, please pay attention. Why are we calling interphase as a resting phase? Why? Because, my lovely kids, no division is taking place. Why are we calling interphase as the resting phase? Because cell is actually not dividing. But do not confuse with this resting phase term. Cell is definitely metabolically quite active during interphase. Please do not forget that. Whenever a cell is in interphase, it is in resting phase because it is not dividing, but it is definitely metabolically active. It is producing or synthesizing DNA molecules. It is replicating the genome. It is producing proteins, RNA molecules required for different functions, right? So metabolically आपकी सेल एक्टिव होगी, बट ये फेज रेस्टिंग फेज कहा जाएगा. क्यों? क्योंकि आपके जो सेल है वो एक्चुअली डिवाइड नहीं कर रही है. सेल ग्रो करेगी, प्रोटीन बनाएगी, आरएनए बनाएगी, डीएनए को रेप्लिकेट करेगी, इस वजह से वो मेटाबॉलिकली एक्टिव है जब वो इंटरफेस में होती है. अब बात करते हैं एम फेज के बारे में. तो सेकेंड जो चीज़ है वो सेल साइकिल के अंदर हम पढ़ते हैं वो एम फेज है. एंड एम फेज इज द मोस्ट ड्रामेटिक फेज. इसे आप मोस्ट ड्रामेटिक फेज भी कहते हैं, मोस्ट ड्रामेटिक फेज. तो क्यों आप इसे मोस्ट ड्रामेटिक फेज कह रहे हैं? क्योंकि जब भी आप एक सेल को एम फेज के टाइम पे देखेंगे, यू कैन सी लॉट ऑफ मूवमेंट, लॉट ऑफ एक्टिविटीज गोइंग ऑन. क्रोमोसोम्स इक्वेटर पे आ रहे होंगे, सेंट्रोमियर विल बी स्प्लिटिंग, क्रोमोसोम्स विल बी मूविंग टुवर्ड्स द पोल. सो लॉट ऑफ ड्रामा, लॉट ऑफ एक्टिविटीज विल बी सीन. एंड एम फेज इज द टाइम, इज द phase of the cell cycle where actually cell will divide. It is further divided into two. First is the karyokinesis and another is the cytokinesis. Now as the name suggests, karyokinesis, karyo का मतलब क्या होता है मेरे प्यारे बच्चों? Karyo simply means nucleus, right? So karyokinesis means first of all nucleus will divide. सबसे पहले आपके न्यूक्लियस का डिवीजन होगा एंड देन आपका साइटोप्लाज्म का डिवीजन होगा. साइटोकाइनेसिस मीन्स साइटोप्लाज्म डिवीजन. Right? So how karyokinesis will take place, my dear students? Karyokinesis, if we are talking about mitosis type of cell division, then karyokinesis will have further stages like prophase, metaphase, anaphase, telophase, and at the end of telophase, the nucleus of the parental cell will split into two daughter nuclei. So during karyokinesis, my dear students, the sister chromatids in a particular chromosomes, my dear students, they will separate out due to splitting in the centromere, and these sister chromatids, after getting separated from each other, they will start acting as a daughter chromosome and will become part of two daughter nuclei. This is how karyokinesis will take place. I will explain you each step, each stage in detail. in coming few minutes. Now, usually, please focus on this word. Usually, karyokinesis is followed by cytokinesis, where the cytoplasm also split and the two daughter nuclei get surrounded by the respective cytoplasm, leading to formation of two daughter cell. So, at the end of cytokinesis, there will be formation of two daughter cell. So, at the end of karyokinesis, क्या मिलेगा? At the end of karyokinesis, you will get two daughter nuclei, and at the end of cytokinesis you will get two daughter cell. At the end you will get two daughter cells. So why am I calling as two daughter cells and two nuclei? Because right now I am considering mitosis type of cell division. So right now you have to consider that this M phase is the mitosis one, right? If you consider it for meiosis, this will change. At the end of cytokinesis, at the end of meiosis, you will get 4 haploid daughter cells. I hope this much is understandable. Now next thing is, now let us discuss about the several phases which lies under interphase. So interphase is also further divided into 3 phases. One is the gap 1 or G1 phase, S phase, and gap 2 or G2 phase. So these are the phases which are under interphase. Remember, interphase में cell क्या करेगा? Cell division के लिए prepare करेगा. So there will be activities that will be happening in G1, S and G2 that will prepare the cell ultimately for division. So let's find out what activities are going to take place. तो सबसे पहले है आपका G1 phase, या जिसे आप first gap period भी कहते हैं. So this is your G1. This is your G. Kind of activities are taking place. First of all, one important fact that G1 is the interval between mitosis and initiation of DNA replication. Up these lines, let me tell you, whatever points are written on the slides, my dear students, most of them are directly from the lines of NCERT, and I have purposefully placed these lines on the slide so that you can understand that NTA is directly making questions by copying and pasting the lines of NCERT. So these are important lines of NCERT that you got to learn, you got to remember, you got to read, observe before sitting into your NEET 2023 exam. These are the most important lines and you got to understand as well. So let me help you out. So it is writing, NCERT is saying that G1 is the interval between mitosis and initiation of DNA replication. So G1 is lying between M phase. Now if you consider M phase as the mitosis one, so this is the M phase, this is the mitotic phase, and hereafter G1 is the S phase. In S phase what will happen? DNA will replicate, right? So that is why NCERT is writing that G1 is the phase between mitosis and DNA replication. Next thing, cell is metabolically active and continues to grow. So there are a lot of activities are taking place, lot of physiological reactions, biochemical reactions are taking place inside a cell when it is in G1. So because of which the contents are increasing, the components of the cell are increasing, the cell is actually growing. Also remember, RNA and proteins required for DNA synthesis. You already have studied about DNA synthesis, how DNA replication takes place in chapter number 6, class 12, molecular basis of inheritance. So you should be aware about this, my dear students, that RNA and proteins are required for DNA synthesis. So in the next phase, which is the S phase, where DNA replication will take place, so the RNA and proteins required for that will be produced in the G1 phase. Next, my dear students, that you need to understand, and here this also many a times you can see question being asked, that most of the cell organelles will duplicate in the G1. Most of the cell organelles will duplicate in the G1. Very important. Now, next phase is the S phase. This is your S phase. ये आपका S phase है, and S phase is characterized by duplication of the genome. That means here the copy of the DNA will be produced. So DNA synthesis or replication will take place. Replication also known as autocatalysis. Next is, my dear students, that you need to understand is during S phase, amount of DNA will increase. Because of DNA replication, amount of the DNA which is there in the cell will be double in comparison to what it was before starting the cell cycle, or you can say amount of the DNA that the cell had when it was in G1. Now the amount of the DNA the cell will have after S will be double that what it had in the G1, right? But not to forget, my lovely kids, that the number of chromosomes will not change. Very important. अब इस fact पे NTA ने several time question पूछा है. Please remember this. Please understand this. That during S phase, amount of the DNA doubles, but the number of chromosome remain same. For example, if this is your parental cell, it has 2n number of chromosome and 2c amount of DNA, right? Before S phase. So this is the condition before S phase. That means you can say in the G1 also, before S phase or in G1, right? Now after S phase, guys, after S phase, the amount of the DNA will become 4c, but the number of chromosomes will remain 2n. तो 2n 2n ही रहेंगे, number of chromosomes same रहेगा, but amount of DNA 2c से 4c हो जाएगा. So amount of DNA doubles, but number of chromosome remains same. Next, in animal cells, my dear students, DNA का replication begins inside the nucleus and centrioles will duplicate into the cytoplasm. इस पे भी NT ने question पूछा है several times. Please remember this. One also very important fact that you need to remember is histone proteins are synthesized during S phase. Next, histone proteins are required for packaging the DNA, double helical DNA into the chromatin format. Next is, my dear students, G2 phase. G2 phase will also synthesize proteins, my dear students, that are required for M phase. So there are proteins, my dear students. For example, you understand in the M phase there will be requirement of formation of the mitotic spindle that is produced with the help of the microtubules. Microtubules are made up of tubulin proteins. So all these proteins will be produced during the G2. The cell growth will continue and even the volume of the nucleus will increase during G2 phase. Pretty simple. Now guys, let us talk about one more important fact, and that is the G0 stage. Now what is the G0 stage? Now listen this carefully. What happens is, there are some cells in an adult animal. Listen this carefully. There are some cells in the adult animals like that of the heart cells or nerve cells which do not divide. And there are many other cells which will divide only occasionally. For example, if there is an injury and a cell or tissue has been lost, in order to replace that, those cells will divide occasionally in the specific condition. So whenever a cell do not wishes to divide or continue the division, my dear students, in that case that particular cell is going to exit the G1 at the end of the G1 and enter into an inactive stage known as G0 or quiescent stage. I repeat myself, my dear students, the cells which do not wishes to continue the division, my dear students, but they can do the division whenever they are called to do so. In that case, these cells will exit the G1 at the end of the G1 and enter into an inactive stage known as quiescent stage, that is the G0. So whenever there is a special condition, whenever there is an occasion, there is a requirement, the cell can again re-enter the cell cycle from G0 in order to undergo division and replace the lost cell, right? So remember this, cells that do not divide further exit the G1 at the end of the G1 to enter in an inactive stage called quiescent stage or G0. The cells, remember, although G0 stage is known as the resting stage, but here the cell is metabolically active. Why are we calling G0 as a resting stage? Because cell is not dividing. But the cells which are there in the G0, they are definitely metabolically active. They will not undergo division until and unless they are called to do so whenever a cell is there in the G0. I hope this much is pretty simple and clear to everyone. Now next thing is we will focus on M phase. As I told you, the M phase is divided into karyokinesis and cytokinesis. So these are the stages of karyokinesis. It will start from prophase, continue to meta, ana and telo. At the end of telophase, you will get the two daughter nuclei, right? Right now we will be focusing on mitosis type of division. So ये जो M phase हम लोग डिस्कस कर रहे हैं वो किसका है मेरे प्यारे बच्चों? ये जो M phase अभी हम लोग डिस्कस कर रहे हैं वो माइटोसिस या माइटोटिक टाइप ऑफ सेल डिवीजन का है, where a parent cell will split into two daughter cell. तो इसमें क्या होगा माइटोटिक डिवीजन में? a parent cell will split into two daughter cells, and both these daughter cells will have right. If a parent cell was a diploid cell, then both the daughter cells will also be diploid. They will have same number of chromosome and same amount of DNA. And if the parent cell is a haploid, even then also mitotic division can take place, especially in the plant cells, my dear students, where a haploid cell undergoes mitotic division to give rise to two haploid daughter cells. So during mitosis, during mitotic type of cell division, the ploidy is maintained, the chromosome number, amount of DNA remains same in the daughter cells, and that is why we call mitosis as an equational division. इसी वजह से आप mitotic division को equational division AKA means also known as equational division. I hope this is pretty simple and clear. Alright, so now let us talk about what are the changes that a cell will undergo when it is under prophase, metaphase, anaphase, telophase. At the end of cytokinesis, you will get the two daughter cells. Let's start with the prophase. Now I want everyone to focus on this diagram. This diagram represent how a cell is going to look like when it is under prophase. Please understand, a cell when it is under prophase, the chromatin material will show condensation, formation of chromatids, and ultimately leading to formation of the chromosome. So this change, the shift of chromatin material, DNA which is in form of chromatin material towards the condensed chromosome, this condensation, this step will take place during prophase. In the entire prophase, my dear students, you can see condensation of chromatin material into chromosome will take place. So here initiation of condensation of chromosomal material into chromosome will start, my dear students. Centro, very important, these are the centrosomes. What are centrosomes? Two perpendicularly arranged centrioles which are surrounded by an amorphous material known as mesiule. So these centrioles, remember, centrioles will duplicate during the S phase in the cytoplasm. So these centrosomes will move towards the opposite end and will start radiating out the microtubules, and these structures will be called as esters. So centrosomes radiate out microtubules called esters. So इस पूरे structure को आप क्या कहेंगे? Esters, right? So whenever somebody is asking you what is a mitotic apparatus, you can say two esters plus spindle fiber. So this is one ester. This is another ester. What is an ester? Centrosomes radiating out the microtubules. This is an ester. There are two esters. They will radiate out microtubules. So a mitotic apparatus is made up of two esters and the spindle fibers which are coming out from there. Next is, my dear students, this is an NCERT diagram. You must remember how does a cell look like when it is in early prophase and when it is in late prophase. So this is how a cell look like when it is in early prophase. In early prophase, you can see the initiation of condensation of chromatin material into chromosome has started, and here you can still see the nuclear membrane. But when we talk about the late prophase, you can easily see that how nuclear membrane has been fragmented. It has converted into fragment. It is not at all continuous. It has changed into fragments, and somewhat you can see the chromosomes becoming more condensed during the late prophase. Always remember one very important point, my dear students, cells at the end of the prophase will not have cell envelope and they will not have nucleolus, Golgi apparatus, my dear students, and endoplasmic reticulum. So cells at the end of prophase do not show Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope. They disappear. Next stage is, my dear students, metaphase. Remember, metaphase. So the complete disintegration of the nuclear envelope that we saw in the late prophase, so it was converting into the fragments. So whenever the nuclear envelope has completely disintegrated, now this This point marks the starting of the metaphase. In metaphase there is a completion of chromosome condensation. इसका मतलब क्रोमोसोम fully condense होकर अब ready है at the start of the metaphase. Nuclear envelope completely disintegrate हो चुकी है at the start of the metaphase. Next, metaphase is the best stage to study morphology of the chromosome, क्योंकि यहाँ पर क्रोमोसोम काफी कॉम्पैक्ट और शॉर्ट होते हैं, जहां आप उसका साइज आराम से और उसको काउंट भी कर सकते हैं. तो क्रोमोसोम का साइज एंड क्रोमोसोम का नंबर, द मॉर्फोलॉजी ऑफ द क्रोमोसोम यू कैन बेस्ट स्टडी इन द स्टेज कॉल्ड मेटाफेज. Next thing is, my dear students, please notice this diagram. In metaphase, the chromosome can easily be seen containing two sister chromatids. तो आपको इस तरीके का नजर आएगा मेरे प्यारे बच्चों, the chromosome. Yeah, so this is a metaphagic chromosome. This is a metaphasic chromosome. This chromosome will have two sister chromatids. तो ये रही आपकी two sister chromatids. ये रही आपकी two sister chromatids, and this is the primary constriction centromere which is holding these two sister chromatids together. If you're confused from where these two sister chromatids have come, do not forget ये दोनों के दोनों जो sister chromatids है मेरे प्यारे बच्चों कहाँ से आई हैं? ये दो sister chromatids इसलिए क्योंकि DNA replicate किया था during S phase, right? So during replication of the DNA, because of the replication of the DNA in the S phase, you have two sister chromatids which are held together with the help of this primary constriction known as centromere. Now this centromere, on the surface of the centromere, there is, my dear students, disc-shaped structures. On the centromere, you can see the disc-shaped structure known as kinetochores. Kinetochores. So what are kinetochores? Kinetochores are the disc-shaped structures which are present on the centromere, and centromere is the structure, primary constriction, which is holding the two sister chromatids. This is how the metaphagic chromosome will look like, and this is how the metaphagic chromosomes will align themselves on the इक्वेटर ऑफ द स्पिंडल. अभी देखो ये स्पिंडल अपैरेटस का इक्वेटर है और इस इक्वेटर के ऊपर सारे के सारे मेटाफेजिक क्रोमोसोम्स आके अरेंज हो गए. उनका सेंट्रोमियर ऑफ ईच ऑफ दीज मेटाफेजिक क्रोमोसोम्स विल बी लाइंग ऑन द इक्वेटर लाइन. दिस इज ऑल्सो नोन एज अ मेटाफेजिक प्लेट. इट इज एन इमेजिनरी प्लेट. तो एज सच कोई प्लेट नहीं है, ये जिस तरीके से अरेंज है सेंटर पर इक्वेटर पे ऐसा लगता है जैसे कि ये प्लेट पर अरेंज्ड है, राइट? सो प्लीज रिमेंबर दिस. So I hope this much is pretty simple and clear. और ये बहुत इंपॉर्टेंट लाइन है जो आपको याद रखनी है. even in the NEET 2023 में इस पे क्वेश्चन आया था. several time we have seen question being formed on that, and in fact NEET 2023 में तो इस पे 2 बार क्वेश्चन आया था. एक बार एज सच पूछा गया था, एक बार मतलब इस पे बेस्ड क्वेश्चन था, और एक बार एक ऑप्शन में, एक क्वेश्चन के ऑप्शन में ये statement था. So it is that important, right? So kinetochores are the site of attachment of spindle fiber. ये जो spindle fibers in aster से निकलते हैं, ये spindle fibers जाकर kinetochores पर अरेंज होते हैं. These spindle fibers that you can see, these spindle fibers go and attach to the kinetochores present on the surface of the centromere. They are the site of attachment of spindle fiber. ये बेहद important line है, इसे नहीं भूलना है, राइट और जब भी ड्यूरिंग मेटाफेज क्या देखने को मिल रहा है, साइटोप्लाज्म के अंदर एकदम अरेंज हो जाते हैं आपके क्रोमोसोम्स ऑन द इक्वेटोरियल प्लेट. प्लीज रिमेंबर दिस. और इसी अरेंजमेंट ऑफ क्रोमोसोम ऑन द इक्वेटर ऑफ द स्पिंडल, माय डियर स्टूडेंट्स, इसी को कहा जाता है क्रोमोसोमल कांग्रेशन. तो कांग्रेशन हो रहा है. कांग्रेशन कब होता है? मेटाफेज में. तब इन द साइटोप्लाज्म ऑन द इक्वेटर द क्रोमोसोम्स आर अरेंज्ड. नेक्स्ट इज माय डियर स्टूडेंट्स एनाफेज. अब एनाफेज को आप कैसे डिफाइन करते हैं? एनाफेज में क्या होगा? माइटोसिस की एनाफेज में देयर विल बी स्प्लिटिंग ऑफ द सेंट्रोमियर. अब देखो मेटाफेज में कैसा था आपका क्रोमोसोम? इफ यू रिमेंबर तो क्रोमोसोम वाज लाइक दिस इन द मेटाफेज. अब आपकी मेटाफेज में अगर क्रोमोसोम ऐसा था तो अब क्या होगा? अब क्या होगा मेरे प्यारे बच्चों? एनाफेज के अंदर स्प्लिटिंग होगी सेंट्रोमियर की. तो ये आपका सेंट्रोमियर है और ये स्प्लिट कर जाएगा. तो सेंट्रोमियर के स्प्लिट कर जाने से क्या होगा? आपके ये जो दो सिस्टर क्रोमैटिड्स थे वो सेपरेट हो जाएंगे. So this is what going to take place in anaphase. In anaphase, because of splitting of the centromere, the two sister chromatids will separate out and start behaving as daughter chromosome. So they will now start behaving as daughter chromosome. Now these daughter chromosomes are different that of the metaphasic chromosome. In what terms? In number of chromatids. तो एक metaphasic chromosome के ऊपर दो sister chromatids थी. There are two sister chromatids. But when we talk about an anaphasic daughter chromosome, it will have only single chromatids. So this is a daughter chromosomes. These are daughter chromosomes. Next. Also remember, this is the best stage to study the shape of the chromosome. तो आपको याद होना चाहिए मेरे प्यारे बच्चों, आपका मेटासेंट्रिक क्रोमोसोम कैसा नजर आता है? V shaped, right? सब मेटासेंट्रिक L shaped, एक्रोसेंट्रिक J shaped, and telocentric I shaped. So these shapes of the chromosome can easily be seen when the chromosomes are in, or when the cell is in anaphase. So it is the best stage. So you can easily see आपके जो splitting होने से आपके जो sister chromatids है, they have separated out, and my dear students, one of the copy or one of the daughter chromosome from each metaphagic chromosome which are splitted into two, one chromosome is moving towards this pole, another is moving towards this pole, and the movement is such as if the centromeres are facing towards the pole and the chromatids are trailing behind. So it appear as if like this. So ये half इधर चले जाएंगे और half ये दूसरे वाले पोल पे आ जाएंगे. Next देखते हैं मेरे प्यारे बच्चों. Next is the telophase. It is the last stage of karyokinesis. Here almost the steps, the reactions, the processes you will see as if they are opposite to that of the prophase. जैसे कि here at the poles, now here anaphase में what was happening, the chromosome started moving towards the pole. So here you can see in towards the telophase, the chromosomes have reached out to the poles. Now at the poles, the chromosome will lose their identity by getting decondensed. They will back convert into the chromatin, my dear students, and on the clusters of these chromatin material there will be formation of nuclear envelope, right? So nuclear envelope developed around the chromosomal clusters, nucleolus, Golgi apparatus, Golgi complex, ER will reform. This is how a cell look like in a telophase. So you can see formation of two daughter nuclei. So this is one daughter nuclei, this is another daughter nuclei. Next and the last stage of M phase is the cytokinesis. So during cytokinesis, cytoplasm will split. Each daughter nuclei will be surrounded by its own cytoplasm, right? So in mitosis type of division, two daughter cells will be produced at the end of cytokinesis, and usually my dear students, cytokinesis take place after karyokinesis, but there are some conditions like that in case of cenocytic condition or syncytium condition where karyokinesis is not followed by cytokinesis. As a result, in a single cell there are multiple nuclei. So syncytium or cenocytic is the condition when karyokinesis is not followed by cytokinesis. As a result, a single cell results into having multiple nuclei. In animal cell, the cytokinesis takes place with the help of furrow formation, and in plant cell through cell plate formation the cytokinesis will take place. These are few important points you got to remember. One more thing that you need to remember over here is, my dear students, during cell division in plant cell, my dear students, during cytokinesis inside a plant cell, what will happen? Middle lamella will be the first wall, cell wall that will be laid. तो सबसे पहले cytokinesis के टाइम पे in a plant cell सबसे पहले जो wall material deposit होता है वो middle lamella होता है, and it is made up of calcium and magnesium pectate. Now just inner to the middle lamella there will be formation of primary cell wall, and when a plant cell is going to mature, inner to the primary cell wall there will be deposition of secondary cell wall material in 2 to 3 layers. But remember, सबसे पहले middle lamella lay किया जाता है, which is a cementing layer which glues or attaches the neighboring plant cells. So that's all I would like to explain about mitosis. Now next thing that we need to discuss about is meiosis. Acha, one more thing, before we start discussing about meiosis, there are few important points that we need to understand about mitosis. In plants, mitosis is observed in both haploid and diploid cell. And also remember, you can expect a question on this. Onion root tip is the best material to observe mitosis because the size of the chromosome is considerable, easily visible, so that you can easily understand or easily observe how mitosis is taking place. So onion root tip is the best material. In animals, mitosis mostly observed in diploid somatic cell, and in some insects like male bee cells also divide via mitosis because male bee cells are actually haploid. So इनको जो next the division also as well as formation of the gametes in a male honey bee, it will take place via mitosis. So that's all my dear students. Now we will start discussing about meiosis. Okay students, now as I said, we will start understanding about meiosis. Till now I have explained about mitosis type of cell division, where the M phase was mitosis. So in mitosis, my dear students, whether it is taking place in a haploid cell or a diploid cell, the ploidy will remain same, amount of DNA, number of chromosome will remain same as that in the parent cell, in the daughter cells. But when we talk about meiosis type of division, it is basically a reductional division, right? What do I mean by that? Meiosis will be taking place in the diploid cell, and the resulting daughter cells at the end of the meiosis will be haploid in nature. So for example, if meiosis is taking place in a cell like this, which is a diploid, So in this diploid cell, there is 2n number of chromosome and 2c amount of DNA. Now when this cell undergoes meiosis, at the end of meiosis, there will be formation of 4 haploid cell. And each of the haploid cell will have n number of chromosomes, c amount of DNA, So this is the situation, right? So this is meiosis forming 4 haploid cells or daughter cells at the end of meiosis, right? So that is why we say during meiosis, chromosome number and amount of DNA reduces to half in the daughter cells. Now next thing that you need to understand over here is, my dear students, the meiosis itself is further divided into 2 types. One is meiosis 1 and another is meiosis 2. Now meiosis 1 will be followed by, or rather I should say, meiosis 1 will include karyokinesis 1 and cytokinesis 1. That means one cycle of nuclear division, one cycle of cytoplasmic division will take place in meiosis 1. Now after meiosis 1, there will be interkinesis, a short-lived stage, and interkinesis will be followed by meiosis 2, where there will be again another cycle of nuclear and cytoplasmic division. So if you notice over here is, my dear students, there is meiosis 1 and meiosis 2, and both will have, my dear students, karyokinesis and cytokinesis. So during meiosis, there are 2 cycles of nuclear division and cytoplasmic division. That means there are 2 cycles of karyokinesis and cytokinesis, but there is a single cycle of DNA replication. So DNA will replicate only once, and that is in the interphase before starting meiosis 1. After completing the DNA replication, after completing the interphase, meiosis 1 will start, and there is no DNA replication between meiosis 1 and meiosis 2. That means there is no DNA replication during interkinesis. ये बहुत ही important fact है और आपका NCERT में दिया हुआ है. Please remember this. Next. Now, as I explained you, my dear students, this is how the whole division of meiosis into meiosis 1 and meiosis 2 look like. So this is your meiosis 1. right? And these are the stages under meiosis 1. So all these stages are karyokinesis. This is karyokinesis 1, right? Meiosis 1 will start after interphase, after DNA has replicated in the S phase. So in meiosis 1, there is first karyokinesis 1 followed by cytokinesis 1. So at the end of karyokinesis, you will get two daughter nuclei, and the daughter nuclei will undergo cytokinesis where cytoplasm will be divided, and as a result there will be formation of two cells known as dyad of cells. Now please understand over here is, my dear students, the prophase. Under karyokinesis 1 there is prophase 1, metaphase 1, anaphase 1 and telophase 1. Specifically the prophase 1 of the karyokinesis 1 is quite elaborate in nature. It is further divided into 5 stages: leptotene, zygotene. Let me show this. All these are the stages of prophase 1. So prophase 1 is further divided into leptotene, zygotene, pectine, diplotene and diakinesis. At the end of the diakinesis, actually prophase 1 ends. So it is quite elaborate, and very important steps and functions and changes will take place inside a cell when a cell is undergoing prophase 1, that I will explain in the coming few minutes. Prophase 1 is followed by metaphase 1, anaphase 1, telophase 1 and finally cytokinesis 1, leading to formation of dyad of cell. Now each of the two cells in the dyad will enter into interkinesis, a short-lived stage, and then each of the two dyad of the cell will enter into, after interkinesis, will enter into meiosis 2. Meiosis 2 also has again another cycle of karyokinesis. Here also karyokinesis will take place. It is known as karyokinesis 2, and these are the stages of karyokinesis 2: prophase 2, metaphase 2, anaphase 2, telophase 2, and finally there is cytokinesis 2, which will lead to formation of tetrad of cell. यानी यहाँ पर आपके 4 haploid daughter cells will be produced. 4 haploid daughter cells will be produced, right, at the end of cytokinesis 2. Now let's study about all these stages in detail. We'll start with meiosis 1, guys. We'll start with meiosis 1. So this is actually my lovely kid, is meiosis 1 का prophase 1. अब prophase 1 का सबसे पहला stage क्या है? Leptotene. And as we already know that prophase actually starts with condensation of the chromatin material into chromosome, और यही होगा, compaction of chromosome continues throughout this stage. This is how the chromatin, this is how the cell will look like. The chromatin is going to get condensed into chromosome. This compaction will continue. Next and very important stage is your zygotene stage. बहुत ध्यान से सुनना मेरे प्यारे बच्चों. तो zygotene में क्या होता है? Two homologous chromosomes will come together and start forming a pair. Now this process of two homologous chromosomes coming together and forming a pair is known as synapsis. So ये जो process है ना, homologous chromosome पास में आते हैं, pair बनाते हैं, इसे ही synapsis कहते हैं. अब ध्यान से सुनना, homologous chromosomes क्या होते हैं? ऐसे दो क्रोमोसोम जो कि शेप, साइज, फॉर्म, स्ट्रक्चर, लोकाई में अगर सिमिलर हैं तो उन क्रोमोसोम्स को आप होमोलोगस क्रोमोसोम कहते हैं. इन अ पेयर ऑफ होमोलोगस क्रोमोसोम, वन यू गेट फ्रॉम फादर एंड अनदर यू गेट फ्रॉम मदर इन फॉर्म ऑफ गैमेट्स, राइट. योर बॉडी इज प्रोड्यूस्ड फ्रॉम अ zygote, a single cell which is formed because of the fusion of male and female gamete, right. So ध्यान से सुनना मेरे प्यारे बच्चों, इस zygote stage में क्या होगा? Two homologous chromosome will come together and they will form synapsis, है ना. इनका पास आना ही synapsis है. They forming a pair of homologous chromosome is synapsis. Now the synapsis is actually accompanied by the formation of a proteinaceous complex which is known as synoptonymal complex. अब ये synoptonymal complex क्या करेगा? इन दोनों homologous chromosome को stabilize करेगा, आपस में साथ में रखेगा. It will make the two homologous chromosomes stay together. So whenever this synoptonymal complex is formed between the synapsed homologous chromosome, this leads to formation of a complex known as bivalent or tetrad. So the two synapsed homologous chromosome is known as bivalent or टेट्राड। यही कुछ चीजें होती हैं, देख लेते हैं हम लोग। दिस इज हाउ द टू होमोलोगस क्रोमोसोम लुक लाइक। दिस इज, लेट्स से दिस इज द होमोलोगस क्रोमोसोम दैट यू गेटिंग फ्रॉम द फादर एंड दिस इज द वन दैट यू गेटिंग फ्रॉम द मदर। शेप, साइज, स्ट्रक्चर, लोकाई, इट विल बी सिमिलर। अब ध्यान देना मेरे प्यारे बच्चों, ईच क्रोमोसोम in a pair of homologous chromosome will have two sister chromatid. ये इसकी sister chromatid है, ये इसके red two sister and yellow will be two sister. But remember, red and yellow will be non-sister. Red and yellow will be non-sister. Or easily you can understand, my dear students, easily you can understand by this thing that whenever, listen this carefully my dear students, whenever the chromatids are attached to the same centromere, whenever the chromatids are attached to the same centromere, they are sister, and whenever they are attached to the two different centromere, they will be non-sister. So one red and one yellow is going to be the non-sister chromatid. So whenever homologous chromosomes start pairing together, this process, this association is known as synapsis. And synapsis is accompanied by the formation of a proteinaceous complex, which is the synaptonymal complex, and as a result the two synapsed homologous chromosomes are known as bivalent or a tetrad. तो ध्यान से देखिएगा, synaptonymal complex क्या करेगा, it will stabilize these two homologous chromosomes so that crossing over can take place easily in the next stage, which is packaging. तो ये है आपका एक bivalent और tetrad. This is what I will say is a bivalent or a tetrad. तो bivalent या फिर tetrad है ये, bivalent, or you can also say it is a tetrad. So what does this bivalent or a tetrad is actually a pair of synapsed homologous chromosome. Pair of synapsed homologous chromosome is bivalent or tetrad. It will have two chromosomes. It will have two chromosomes. और इसके साथ साथ में इसके अंदर क्या होंगे, it will have two centromeres. दो क्रोमोसोम्स हैं, दो सेंट्रोमियर्स हैं मेरे प्यारे बच्चों, और क्या है, 4 chromatids है, because each chromosome will have 2 chromatids. So there are 2 chromosomes, 2 centromeres, but 4 chromatids. Please remember this. Now in the next stage, which is the Pachytene stage, these bivalents, these tetrads will become more visible, clearly visible my dear students, and this Pachytene will be characterized by the appearance of recombination nodule. अब ये recombination nodule क्या होता है मेरे प्यारे बच्चों, recombination nodule वो site है जहां पर crossing over होती है. So recombination nodule is the site at which crossing over CO occurs. So recombination nodules will be appearing between the synapsed or pair of homologous chromosome. So in synapsed homologous chromosomes के बीच में recombination nodule दिखने लगेंगे, and these recombination nodules are the site where crossing over occurs or take place. अब ध्यान से सुनना मेरे प्यारे बच्चों, इस पैकेटीन स्टेज में क्या होगा, क्रॉसिंग ओवर होगा. क्रॉसिंग ओवर मीन्स एक्सचेंज ऑफ जेनेटिक मटीरियल. व्हाट इज क्रॉसिंग ओवर? Crossing over means exchange of genetic material. So what is crossing over, guys? Crossing over means exchange of genetic material. So genetic material will be exchanged, my dear students, between two non-sister chromatids in a pair of homologous chromosome. So एक homologous chromosome का पेयर की अगर मैं बात करूं तो in a pair of synapsed homologous chromosome between the two non-sister chromatids there will be crossing over. तो ये रहा आपका pair of synapsed homologous chromosome. This red and this yellow, they are non-sister. They are red and this yellow, they are non-sister chromatids. Now between these non-sister chromatids in a pair of synapsed homologous chromosome, crossing over will take place. So you can see in this red chromatid there's a portion of yellow, and in the yellow chromatid there's a portion of red. That means red portion, a part of red chromatid has been exchanged with a part of yellow chromatid. So there is exchange of genetic material. This is crossing over which is taking place in Pachytene stage at the recombination nodule site, and this crossing over will leads to, my lovely kid, this crossing over will leads to recombination. This crossing over will leads to recombination. recombination of genetic material. Recombination of genetic material. क्रॉसिंग ओवर से जेनेटिक मटेरियल रिकम्बाइन हो जाएगा. सो इस टाइप के जो सेल्स बनेंगे, डॉटर सेल्स बनेंगे, दे विल नॉट बी एग्जैक्ट रेप्लिका ऑफ द पेरेंट सेल, एंड व्हेन सच सेल विल पार्टिसिपेट in formation of a new organism, when such cell will act as a gamete, when they will fuse to form zygote, and that zygote will develop into a new organism, such organism will not be an identical or won't be a replica of the parent organism. So this crossing over, which is taking place in the Pachytene stage of the prophase 1, is responsible for recombination, responsible for variation. It is a very important step as far as your evolution process is concerned. Now crossing over is an enzyme mediated phenomenon, my dear students. Enzyme recombinase is required for crossing over to take place. Next stage is the diplotin stage. Now listen this very carefully, my lovely kid. The crossing over has already been completed. So it was initiated as well as completed in the Pachytene stage. Now listen this carefully. Now these homologous chromosomes, these synapsed homologous chromosomes are not required to stay together. So the dissolution of synoptonymal complex will take place. So diplotin is marked by or recognized by dissolution of synoptonymal complex. So the synoptonymal complex that was holding these homologous chromosomes together, it will dissolve. So in diplotin, the homologous chromosome will start separating. Remember and notice this very clearly, my dear students. I am repeating for you again. So the separation of homologous chromosome will start from diplotin stage. These homologous chromosomes came together during the zygotine stage. They will start separating from the diplotin due to dissolution of the synoptonymal complex. But still these homologous chromosomes will be attached to each other at the sites where crossing over took place. Now these sites will be observed as egg-shaped structures known as chiasmata. So what are chiasmatas? Chiasmatas are the site where crossing over took place, and these homologous chromosomes will still be connected to each other at these chiasmata even after the dissolution of the synoptonymal complex. Very, very important. So these are the chiasmata where these homologous chromosomes are still connected to each other. Now these chiasmatas or let me tell you, my dear students, these chiasmata I already explained you, they are egg-shaped structures. When you see it, this is how the chiasmata look like. So in oocyte of some vertebrates, diplotin can last for months or years. This is an important fact given in NCERT, so we got to remember it. Next is diakinesis. It is the last stage of prophase 1. Now this diakinesis is marked by terminalization of chiasmata. So chiasmata will start moving towards the poles. It might even slip off or might remain at the ends. So these homologous chromosomes will still be staying connected with the help of terminalized chiasmata. Also, the chromosomes will be fully condensed at the end of the prophase 1, because diakinesis is the last stage of the prophase 1. Meiotic spindle will start getting assembled in order to prepare these homologous chromosomes to separate finally. The separation of homologous chromosomes started in diplotin, but it will finally be completed in the anaphase 1. Also, nucleolus, Golgi apparatus, my dear students, nuclear envelope, they will start disappearing, or nuclear envelope will break down. Next is, my dear students, metaphase 1. So what is going to happen in metaphase 1? The bivalents will start arranging themselves in the cytoplasm on the equatorial plate. So congregation of the chromosomes will take place. This is how the bivalents, two homologous chromosomes which are still connected to each other with the help of terminalized chiasmata, they will start arranging themselves on the equatorial plate, right? This is chromosomal congregation. Now with the help of microtubules which are coming from the opposite poles, my dear students, these microtubules will be attached to the kinetochores present on the centromeres of these homologous pair of chromosomes. So this is how the centromere with kinetochore, and on these kinetochores the microtubules are coming and attaching. So this is how the bivalents are arranged on the equatorial plate, metaphagic plate. Next is, my dear students, anaphase 1. Please remember what will happen in anaphase 1. Because of the shortening of the microtubules, the two homologous chromosomes will finally separate out. But remember, centromere will not split. There is no splitting of the centromere, so the sister chromatids will not separate. The sister chromatids will still be together because splitting in the centromere has not taken place. Only homologous chromosome will separate out. One will move towards this pole and another from the pair is going to move towards this pole. So this is how in anaphase 1 the separation of homologous chromosome finally completes, and one from each pair will reach to the one pole and another will to the another pole. Once the chromosome have reached towards the pole, what will happen? The telophase 1 is going to start, the nuclear membrane and nucleolus will reappear and cytokinesis will follow, and as a result dyad of cell. So after telophase 1, what will happen? The cytokinesis 1 will take place, and as a result, my dear students, the dyad of cell at the end of cytokinesis, you will say. So at the end of cytokinesis 1, there will be formation of dyad of cells, right? So in telophase 1, nuclear membrane, nucleolus reappearing, and the cytokinesis is followed, forming the dyad of cell. Now what will happen? Interkinesis. Both these two cells, dyad of cell, will enter into a short-lived stage known as interkinesis. This stage is between the two meiotic divisions. So between meiosis 1 and meiosis 2, there is interkinesis. DNA will not replicate. Alright, and this will be followed by interkinesis, will be followed by prophase 2. Remember, my dear students, meiosis 2 is similar to that of the mitosis. The events will be similar, and during this, my dear students, centromere will split. So in anaphase 1, centromere did not split it, but in anaphase 2 of meiosis 2, centromere will split, and this meiosis 2 is simpler in comparison to meiosis 1. So prophase 2, very simple, nuclear membrane, nucleolus will again disappear, chromosome again become compact, and formation of spindle will start so that centromere can split and daughter chromosomes can be produced. So this is how, my dear students, these are the two dyad of cell, and this is how these two dyad of the cells are going to look when they are in prophase 2. So you can see this nicely over here, nuclear membrane disappearing, chromosome becoming compact, formation of spindle starts. So with the diagrams, you can easily remember these steps. You don't have to cram them. Next is metaphase 2. So these chromosomes will arrange themselves in the cytoplasm on the equatorial plate. So this is how the chromosomes will be attached, and the spindle fibers will attach to the kinetochores present on the centromeres of these chromosomes. Remember, each chromosome of the metaphase will be made up of two sister chromatids. Every chromosome is having two sister chromatids, right? Please remember this. Now, please remember that in anaphase 2, what is going to happen? Splitting of the centromere. This is metaphase. Guys, this is metaphase 2. ये आपका metaphase 2 है और ये आपका anaphase 2 है. तो anaphase 2 के अंदर क्या हो रहा है मेरे प्यारे बच्चों? मेटाफेज से जब एनाफेज की तरफ हम मूव कर रहे हैं तो देयर इज स्प्लिटिंग ऑफ सेंट्रोमियर. एज अ रिजल्ट ऑफ स्प्लिटिंग ऑफ द सेंट्रोमियर, the two sister chromatids are becoming separate and they start behaving as daughter chromosome. So these are nothing but your daughter chromosome, and each of the daughter chromosome, my lovely kid, will be made up of one chromatid, right? Half will move towards this pole and half to this pole. Finally, in telophase 2, what will happen? The chromosomes that have moved toward the poles, they will uncoil and form the chromatin net. Two groups of the chromosome will get enclosed by the nuclear envelope, and finally there will be formation of a 4 haploid nuclei. So you can see there is a formation of 4 haploid nuclei. Each nuclei of each of the dyad of the cell will split into 2 nuclei. So there is formation of 4 haploid nuclei. Telophase 2 is going to be followed by cytokinesis 2. As a result, there will be formation of 4 haploid daughter cells at the end of the cytokinesis too. So this is how, my dear students, in meiosis, one diploid parental cell finally divides and give rise to 4 haploid daughter cell. Now let's talk about the significance of meiosis and mitosis. Now meiosis is responsible or helping in conservation of the specific chromosome number in each species, right? So if we talk about animals, if we talk about humans, we are sexually reproduce. Generally the organism, the higher organism are sexually reproducing. That means in them the new organism is produced with the help of fusion between male and female gametes. Now gametes are haploid. Such haploid gametes are produced via meiosis in the gamete mother cell. And when haploid gamete fuses to produce a diploid zygote, the chromosome number is maintained. The parent organism is having diploid cell. Again, the zygote from which the new organism will develop, it will also have diploid cell because of this meiosis phenomenon. Now meiosis increases the genetic variability because remember in the meiosis 1 in prophase 1 Pachytene stage there is crossing over leading to recombination of genetic material leading to variation, genetic variability in the population of an organism from generation to generation, which is very important step in the process of evolution. I already explained this point. So these are two important significance of meiosis. Now let's talk about the significance of mitosis. So mitosis, whenever an organism need to produce diploid cells, or rather I can say whenever an organism needs to produce cells which are identical to the parental cell, whenever a diploid organism needs to produce diploid cell or a haploid cell need to produce another haploid cells, whenever the ploidy needs to be maintained, my dear students, mitosis can take place. So it result in the production of diploid daughter cells with identical genetic material. So whenever identical daughter cells need to be produced, we can go with the mitosis. That is why mitosis is used in the growth of multicellular organism. Any organism, sexually reproducing organism, will be produced via several mitotic division in the zygote, right? So growth of a multicellular organism will take place via mitosis. Even repair, if cell degenerates, get damaged, my dear students, even that cell can be repaired or replaced with the help of mitosis. So this is all about your significance of meiosis and mitosis, and this actually finishes your chapter cell cycle and cell division. Thank you for listening. Do consider giving me a thumbs up. Do let me know in the comment section how you found this lecture, and remember this chapter, from this chapter 4 questions at least will be asked in your coming NEET 2023. All the very best. Do not forget to hit the bell icon so that you will not miss out any lecture in future. Bye bye. Take care. Thank you.